Welcome to today's webinar. I'm Catherine Dold with CNEN, and I'll be moderating today's event. This webinar is titled The Predict First Paradigm, How Digital Chemistry is Changing Drug Discovery, and is being sponsored by Schrodinger. CNEN works with sponsors to identify topics of interest and value to CNEN's audience and consistent with their mission to provide news and analysis of the chemistry enterprise in a timely, accurate, and balanced fashion. During the webinar, you can adjust the size of the slides on your screen by grabbing the lower right corner with your mouse. If you need technical assistance, please look for the Help widget at the bottom of the screen or type your query into the Q&A box. If you get disconnected during the webcast, please log back in according to the instructions you received earlier. You're encouraged to contribute to the success of this webinar by asking questions at any time during the presentation through the Q&A box on your screen. Our speakers will answer them at the end of the presentation, and as your moderator, I'll be posing as many as time permits. Any questions that we are not able to get to in today's broadcast will be answered by email after the event. Please note that CNEN does not endorse any company, products, or services that may be mentioned in the webinar, and that the webinar will be archived at CNN Online after the live webcast. Our presentation today is being sponsored by Schrodinger. Schrodinger is transforming the way therapeutics and materials are discovered. Schrodinger has pioneered a physics-based computational platform that enables discovery of high-quality, novel molecules for drug development and materials applications more rapidly and at lower cost compared to traditional methods. The software platform is licensed by biopharmaceutical and industrial companies, academic institutions, and government laboratories around the world. Schrodinger's multidisciplinary drug discovery team also leverages the software platform to advance a portfolio of collaborative and proprietary programs to address unmet medical needs. Our speakers today, all from Schrodinger, are Wade Miller, a senior scientist on the education team, Olivia Lines, a strategic deployment manager, Eugene Hickey, a senior research leader in the therapeutics group, and Alice Hooper, a senior scientist also with the therapeutics group. I'll now hand the program over to our first speaker.
Hello, everyone. Welcome to today's webinar, where we'll introduce a Predict First paradigm and discuss how digital chemistry is changing drug discovery. My name is Wade Miller. I'm a member of Schrodinger's education team, and I'm going to pass it off to my other colleagues who will be introducing themselves.
Hi, I'm Olivia Lines, and I'm a member of the Enterprise Informatics team within Schrodinger and work primarily with live design.
Hi, I'm Alice Hooper. I'm a senior scientist in the therapeutics group in Schrodinger, and I'm a medicinal chemist.
Hi, I'm Gene Hickey, a senior research leader in the Schrodinger therapeutics group. I was originally trained as a synthetic chemist, but I've transitioned into a computational chemist, spurred by my interest in the field. I've been working at, I started my career at Boehringer Ingelheim in 1995. As a medicinal chemist, and worked there for 7 years on the bench. In the second half of my career, for the next 15 years, I worked as was invited to join the computational chemistry group, and I worked there. And during my early years at BI, I looked at tools very broadly, basically. But once I joined the computational chemistry group, I got into more advanced techniques and strategies, including pharmacophore modeling, homology modeling, cheminformatics, and some use of software, BI Builder and BI Claim, which were at the time large-scale enumeration and virtual screen tools, which were very good. But when I moved to Schrodinger, I got into and I started learning more about physics-based tools. Now, FEP is amazing. FEP is free energy perturbations, which measuring the binding affinities between ligands and proteins. And this is a physics-based approach. Also, other physics-based models that we have can predict solubility and permeability. We also have expanded from running FEP to large-scale FEP. where we're using the FEP as a training set for machine learning models, where then we can analyze millions and even billions of compounds very quickly. The appearance of machine learning in our workflows has really, I guess, catalyzed my way of thinking about programs from what I call my de novo 1.0 design days to my 2.0, where now I've got the ability to not only look at docking, but more advanced FEP, but also looking at a lot of different properties and machine learning predictions all at once. There's also more availability of structures like cryo-EM and AlphaFold, which have certainly advanced the field as well. One of the biggest differences between my de novo 1.0 and de novo 2.0 days is the culture of collaboration. Now, back in 1995, I was working either by myself or with chemists one-on-one or one-on-two to get ideas incorporated into, get modeling ideas incorporated into the project team. Now in 2017, moving to Schrodinger, I was introduced to this web-based enterprise informatics platform we call Live Design. Now, this is a web-based tool where teams of people can collaborate and ideate new compound designs together from anywhere in the world. So you can think of it like working in Microsoft Word or Google Docs, right? When you're in MS Word, you're working by yourself. You're editing the document and you're sending it off to a colleague for their review. Whereas in Google Docs, you're working together collaboratively because it's web-based. And that's kind of like what Live Design is. It's a collaborative platform where we can all work together. So I'm really enjoying having the ability to predict binding potencies, to evaluate virtual compounds with machine learning and physics-based approaches before synthesizing compounds. So how is it that we can do this? Well, we've got certainly improvements in the computing hardware and the GPUs. So we're getting faster results to meet the timelines of the project. And this is very important. We've also got improvements in force fields and algorithms, which make the predictive models more accurate. And based on this, or using this web-based informatics infrastructure, this Live Design, we're able to have the predictive models more accessible across all people in the team and available also in our daily workflows. So here's our first snapshot into Live Design. So what you can see from this limited view is some of the features that we love about Live Design. So in the first column, you see the structure. This can be from a two-dimensional hand-drawn structure within Live Design. There's enumeration tools within Live Design to populate the different structures, or you can import them from external places. The next column highlighted, we've got the lab assay. So if their compound has been made and tested, we can incorporate experimental data. But before we have that, we look at a lot of different predicted models. For example, I'm just showing here the docking model. So you run the docking with a click, and you get the docking score. You get a 3D representation, which if you click on that 3D methane, you get the window to the right, where now you can spin the molecule in 3 dimensions. You also see the red and green balls in there. This is from WaterMap, another software product that we use. So a lot of these things can be incorporated into how we evaluate our designs. The predicted properties that you don't see each have their own column, and they could be things like physical chemical properties, potency, selectivity, some of the physics-based approaches. We have the FEP, permeability, solubility. So any model that you can imagine, and you have the ability to build machine learning-wise, can be incorporated and used within Live Design. Now we talk about this, let's say, a predict-first paradigm, where we want to predict, let's say, use all of these models to predict our compounds, to evaluate our compounds, to predict which one should be made first. We talk about DMTA cycles as medicinal chemists, right? Design, make, test, analyze. And we can do this in Live Design too. We can do this in silico in Live Design, right? To identify the best compounds for synthesis. And after doing this in silico, we can use the experimental DMTA cycle to make the compounds, get them analyzed experimentally. And watch the whole entire process repeat. So live design gives us a platform for this large scale exploration and to analyze all of our data and to bring it all together. Okay, so I want to introduce you to the Schrodinger Therapeutics Group. So we're a full internal drug discovery team made up of medicinal chemists, computational chemists, biologists, preclinical and clinical development teams as well. We work in collaboration with others, and we also have our own fully owned pipeline as well. We deploy a digital chemistry strategy. So with using our large scale physics-based approaches, machine learning models, and also the live design, the collaborative enterprise informatics system. And we focus on not only structurally enabled programs, but also structurally enabling programs. So that's something new that we're doing as well. So we're using X-ray structures, cryo-EM, and if we can, homology models. Now our goal is to perform fast in silico design, predict, analyze cycles. And you can see this on the picture on the right in the blue circular arrows. We then triage down to only a handful of compounds for the full DMTA cycle, where we can make and test them. So our therapeutics group has been working on this for many years, and we've got a large pipeline of programs. I'm showing you a few of them on this slide: ACC, TYK2, alpha-4, beta-7. We strive for best-in-class, but also first-in-class programs across a diverse target classes. We've also worked to collaborate and co-found different companies as well. Nimbus and Morphic are two examples. Both of these are exciting companies for us because they've both released phase 2 data, positive phase 2 data very recently for compounds that were actually designed in collaboration with Schrodinger. So it's been a very exciting 6 years for me here at Schrodinger. I want to introduce next Alice Hooper, who will tell you more about how we use live design.
Thanks, Stu. Here at Schrodinger, we use live design every day as a key collaborative tool for all disciplines, as Jean mentioned. From medicinal chemists adding designs and checking properties and results, both experimental and predicted, to running our own docking and machine learning models, which are constantly updated and validated by our computational chemists. Our computational chemists also contribute designs and upload the latest predicted binding affinity, permeability, or solubility, whichever model that project is focused on utilizing. Notice that the ratio between computational chemists and medicinal chemists here at Schrodinger is higher than you would expect to see elsewhere. And that is to ensure that the computational chemists have the bandwidth and the time to dedicate to each project. In addition, our biologists and DMPK teams are able to use live design to update experimental data for everyone to view and utilize various different ways of visualizing this data. Data that is then used to further validate our models. All of which makes it simple for our leadership and collaborators to get an effective and efficient picture of the progress of a project at just a quick glance if needed. Another way that we're utilizing live design is in design clubs. These are organized sessions where a project team discusses a potential issue with a chemical series, be it potency, selectivity, or ADME. And we have a forum inviting medicinal chemists from across the time zones and project teams, and those computational chemists that wish to join, to attend and design to try and help combat this issue. It allows the project team to see and analyze the bigger picture through active discussions and hundreds of diverse ideas generated in just 1 hour session. In many cases, a number of leading chemical series were originated from or inspired by ideas from outside the immediate project team. We're hoping to give you a taste of how these design clubs work in a live demo later on in the webinar. I'll hand over to my colleague Olivia.
Great, thanks, Alice. So before we get to the design club portion of this webinar, I'm just going to highlight some of the capabilities of live design and some of the tools that you would have available to you. So as has already been mentioned, Live Design is a single platform where users from medicinal chemists to computational chemists to project managers can all access the same set of data. It provides a way for you to integrate both your lab data, your SAR analysis, and your 3D modeling data into one easy access place. We've got SAR tools available, such as R-group and reaction enumeration. So these can be used on not just the synthesized and registered compounds in your organization, but also in your design ideas as well. You can examine both your real, so that's your synthesized, computationally or experimentally compounds, and also any design ideas you have in a single live report or a single place within Live Design. One such tool that we have available within Live Design is Ligand Designer. So Ligand Designer is available now through the Live Design interface. It provides a 2D or 3D design space for you to ideate and explore a ligand that is part of a ligand receptor complex. It allows you to gain different information about the ligand receptor complex via these 3D overlays, which can allow you to explore these interaction potentials that you can see on the screen. It allows you to examine the growth space. So you can see here, we've got this different shading with the different color blues, and you can also examine the receptor surface. Through Ligand Designer, you can also incorporate the water map overlays and also the sitemap overlay. So we'll give you a bit more of an introduction into Ligand Designer in the Design Club later, but for now, this is just a taster of what it looks like. Within Live Design, you also have the ability to set up MPO scoring. So these are your multi-parameter optimization scoring functions, and this allows you to assess, does a compound fall within a desired property range? So MPOs are individual scoring functions which can be generated from a combination of experimental and computational parameters and allows you to answer that question. Each parameter can be set on the curve of your choosing. So is the middle value better or a higher value better? And then each parameter is weighted before being combined into a single MPO score. So you can see over here on the screenshot where we have this add me MPO, that my values run between 0 and 1, and we have automatic color coding in place on live reports. Another key function of Live Design is the ability to access your data from within the UI. And through that, I want to introduce to you the Assay Viewer tool. So the Assay Viewer tool collates all of the assay data available for each compound across the entire project and allows you to see what is available that's associated with this compound beyond what you have already included within your live report. It allows you to further your design process by drawing information from different places and perhaps highlights to you that other experimental or computational information has been gathered on this compound that you weren't aware of before. And the assay tool is available through the Live Design UI. Finally, I want to introduce to you our dashboard concept. So through Live Design forms, you can use dashboards to effectively collaborate and triage different compounds with your colleagues using a set of physiochemical or multidimensional information that you have gathered into one place. So now I'll just take you through two separate setups that we have. The first of these is the decision to support dashboards. So this is on the screen is a typical dashboard that I see users rely on in their workflow regularly when triaging individual compounds for further investigation. You can see at the top, I have both the 2D and the 3D rendering of the compound image. In the middle, I have some PIC50 data, as well as some information on the docking jobs that have been run for this particular compound. Now, a user can use this information to inform what it is they would like to request next in the design process. So you can see down here at the bottom that we have different fields where we can request things like solubility or SPR. In this compound's example, we've requested all of that information, and perhaps the user is just coming back to check that all of the information has been collated into one place. The second dashboard that I would like to introduce to you is our hit follow-up dashboard. So this dashboard has been set up more as a plot view. You can see right in the center that we've got this plot of PIC50 against the clusters. We've got curves in the bottom corner, as well as a radar plot of various different calculated properties on the bottom right. So all of these fields are interlinked. So if you click on one particular portion of data in one plot, it will highlight the relevant data in all the other plots. We'll show you more of how these dashboards work in our design club later, but I do want to highlight that dashboards such as these are just created by the everyday users of live design. There is nothing overly complex about creating them, and they can be created by anybody. Just to summarize my portion, what I've shown you today are just a few key tools that are available within live design to allow you both to design and manage your way through your entire DMTA cycle. Within live design, we're examining live data, live model execution, and giving you the most up-to-date decision-making within your organization. I'll now hand over to Wade to talk you through the design club portion. Thanks.
So as Alice and Olivia mentioned, we'd like to give you a little bit of a taste of what a design club actually could look like. So in this particular example, we're going to be working off of a recent paper in DMedChem, from a Schrodinger and Takeda collaboration, where they focus on designing a novel class of D-amino acid oxidase inhibitors. In this case, they use a tool called Auto Designer along with human designs, fed all that information into live design and use that to triage the compounds and progress the project. So now we figured, following along with what Olivia just mentioned, the best way to actually introduce this project is with a dashboard in live design itself. So what you can see over here is a forms view within live design that is set as a dashboard to really summarize the information at this point in the project. So just to orient you a little bit, so these are various compounds here. You can see that we have our carbon linked and then our sulfur linkers at different positions. And all the information below it is corresponding to the data on top. It is important to note, this is all entirely customizable. None of this is, you aren't sort of forced to do any of this in any way. So we can make whatever type of analyses we'd like for whatever is helpful at a particular stage in the project and for whatever task you're trying to do. So in this case, we have some predicted and experimental potency related data. We have some predicted and experimental pKa data. We have some predicted ADME data. And these are from various models that are integrated into live design, whether the Merck CNS PMPO or an ML model trained on log-D data. And then we also have, in this case, some experimental PK data as well. And in order to motivate this design club, I do want to mention, so in this case, as you can see, we have 3 compounds right here, all with different linkers. And what the researchers working on this project had identified was that these sulfur-linked compounds were able to lower the pK of the head group compared to the carbon-linked analogs, which then allowed it to favor the bioactive tautomer and really helped sort of shift the tautomer distribution in a desired direction. And we can see this right over here. So in this case, we have our carbon-linked compound right here, where our predicted pK was 9.65. And then here where we have the sulfur linker, we have our experimental pK at 8.52. So we were able to, in fact, drive this down. And we can see that with several other mass molecular pairs in this project. And the goal that we're going to be simulating here that they actually did in this project is to explore, while viewing these 2 different sulfur-linked scaffolds, let's explore some para and ortho substitutions on the phenyl group, potentially single substitutions, potentially simultaneous substitutions. And before we actually jump in there, I do want to show over here. So this is sort of synced. This is a plot here, part of this forms view dashboard, synced to all the data that is available in this project at this point, where we're sort of plotting our binding efficiency MPO that we set using those cutoffs, where we're able to customly set using cutoffs as Olivia showed before, and then ADME MPO that's bringing in some of our predicted data that we have. And we have these colored, so our 3 reference compounds are colored right here. So this is our carbon linked, and then our 2 sulfur linked compounds, as we can see, are quite similar right here. So what we're going to do is actually design in parallel and feed all of those together. So this live report, which Olivia is going to show a bit later, is where all of our designs are going to be funneled into. So we're going to be able to gather all of the designs that we're doing and pull them into one single place where we could then go through more, perhaps more extensive analysis. So to start off a little bit, I do want to then actually show what are we all working with. So if we're in a design club, a big portion of this is, well, designing. So within this, we have our 2 reference compounds. So these are frozen in this live report. We have some predicted properties. So these predicted properties and all the models here are set to run automatically. The second a new design is added to this live report, these jobs will be kicked off immediately. So you have these predictive models. We have docking models. So no need to reach out to a colleague and ask them, can you dock this compound for me? It's done right away, automatically, using a well-validated docking protocol. We then have water map data that both Jean and Olivia alluded to earlier on. And then, as mentioned in our dashboard before, we have our predicted PKI data, ligand efficiency based on that, our ML predicted log D, and then our property forecast or solubility forecast index that we're going to be using as some of our optimization goals in this project, as well as the Merck CNS PMPO, and some information about how the compounds that we are designing compare to patented DAO inhibitors that we were able to find. So to perhaps show an example of what this design actually looks like, I want to sort of walk you through it. So I can simply take a compound from here, drag it into the sketcher, and I could quickly ask, what if I change that chlorine to a fluorine? Add that idea to my live report, and I get that near immediate feedback on these various properties. So I could see my PSA, molecular weight, hydrogen bond donors, hydrogen bond acceptors, and then I could very quickly go and look at my this doc compound. I can overlay it with my reference and quickly see, is there any difference that we're seeing, which we wouldn't necessarily expect with something like this. And then I can also overlay it with that water map data to see if I'm potentially, with such a small change, we wouldn't expect any big difference, but whether I'm building in a direction where I might be able to displace a high energy water and get some of that thermodynamic reward by displacing those high energy waters in the pocket. So in addition, and let me just unselect this reference. So in addition to doing this more standard 2D design, I also wanted to show the type of design that Olivia alluded to as well. So in this case, I can click edit right here. And I can initialize ligand designer. And I do want to know at this point, as I'm doing some designing, my colleagues are also designing in their own live reports. So this truly is distributed design where we are designing separately and funneling these ideas together. So in ligand designer, and I'm able to decide how I want this information to be displayed. Do I want it to be, do I want to look at the inverse pharmacophores in the protein, see what potential interactions might exist? Do I want to overlay with the water map? It's really whatever information will help you as you're going through your design process, you can load in, or if information is maybe not too helpful for you and actually makes it a little clutters the workspace, you can toggle it off. So it really is your own sort of custom design workspace. And what Ligand Designer allows you to do is to design in either 2D or 3D. So whatever you are most comfortable with, whichever is most intuitive for you at the particular point you're working on a project, and gives you also incredibly quick feedback. So let's say I want to move this to change this to a carbon, and then add 2 fluorines there. And then if I click predict pose, I'll pretty much instantaneously get my new pose predicted right here. And then I could use this as a way to pre-triage my compounds and then decide whether I would like it to add it to my live report or not, whether it's something I would want to share with my colleagues. It's a really great sanity check as you're walking through projects to see, will this reasonably fit in the pocket, yes or no, and then decide what to do with it afterwards. So I can click add selected ideas right here. And if I clicked on editing, we can see that it has been added to my live report. And some of these models, as you can see, are kicking off immediately because we're going through this process of designing these new compounds and really using these computational models to assess them in real time. I'd now like to hand it over to my colleague, Olivia, who's going to be taking it from here.
Okay, great, thank you. So you can see on the screen here, I'm sharing the same live report that Wade was sharing, but while he was talking, we've been adding some different design ideas into this live report from our own collaborative live reports. Before we start talking about why we're making the designs that we're making, I just want to emphasize the huge collaboration aspect of this tool is that I'm currently sat in the UK, Wade is currently in Portland, and Alice and Jean are currently in New York. We're all accessing the same server, we're all looking at the same data, and we're all designing in real time. Okay, so let's take a look at a few of the compounds that have been designed. So if I just open this up in 3D, then we can get everybody else to start designing in the background. And perhaps, guys, if you want to chime in with any comments as to why you are designing what you're designing. So if I look at this ligand now in 3D, and if I just turn on the pi stacking interactions and zoom in, then you can see here how this head group is quite structured in the pocket here, but really what we want to be doing is designing into this kind of region over here. And you can see if I overlay one of the initial structures into this 3D model, that actually they're all still aligned in the same way. We've just added this group on the end. And I can do that with any of the designs as they come into this live report. So I can just highlight here at the bottom, we've now got 31 compounds total in this live report, but 10 that we're actually interested in looking at and comparing to our starting structures over here in the top. Great. So if I switch back to designing into my own Design Club live report at this point, I'm going to drag over this compound into the sketcher, and perhaps actually I want to add something over in, sorry, I've lost the orientation, that's in 2D. So I just want to extend this part of the molecule over here. So I'm going to just draw a longer chain, add that idea to my live report, look at all of those new molecules, new models that are being calculated. You can see these gray squares here flashing, and then hopefully that will be added into my Design Club overview as well. Alice or Jean, are there any particular compounds you'd like me to talk through at this point that you've added to this live report? If you just shout out the compound number, then I'll get it up on screen.
I've got one for you.
Yep.
All right, how about V131553?
Right, if I just find that in the live report. So I can highlight that compound there. And if I just look at it in 3D, do you want us chat us through why you designed this particular compound?
Sure, yeah, I saw some available hydrogen bonds out in this area, a donor and an acceptor. And I thought that this alcohol out there would fit between them, and I was able to access that with an ethyl bridge between the two oxygens.
Okay, great. Right, I'm going to switch to our dashboard now and see how our data is coming along. So we've got this Design Club overview dashboard. You can see that this is being automatically populated as data is coming in. So just to explain what's going on here in our dashboard, over here on the left-hand side, we've got our property forecast index using the predicted PKI and predicted log P. So what we're caring about in this particular plot is what's appearing here in the top left quadrant. And you can see as I'm selecting these points on my graph, that's automatically populating these tiles over here on the right-hand side. So these tiles just give you a drill down into the data associated with the compounds that you selected. So you can see I've got the predictive properties at the top. I've got my, and then I've got my DAO patent search similarity score, which is telling me how unique these compounds are against a patent search. And I can scroll down here and look at all of the compounds that I have selected. And as I deselect, it also removes them from the tile view. In the bottom right, we've just got a box plot. So this box plot is allowing us to look at a comparison of where the sulfur is in the linker. So you can see here that actually the distribution of values is not particularly sensitive to the sulfur location, and we can consider both of these moving forward in the design cycle. So within a forms view, I'm also able to filter this down based on other information that I've got. So I've got some filters set up, and I can look at a single match scaffold by itself. And again, I can select some compounds here and consider maybe that adding things into that particular position in the ring haven't increased the score in the way that maybe I was hoping. And I can look at in each individual match scaffold, or I can turn those filters off and look at them both in the same way. So at this point, I'm just going to select the compounds that I might be interested in, and then I'm going to filter the live report to the selected compounds that I'm interested in. You can see here that my plot has automatically resized, and then if I switch back into spreadsheet view, it's automatically selected just those 18 compounds that I am interested in. And then I'm free to click around the data as I would be in any normal way. I can open up the 3D viewer and can compare each of these to the starting structures and overlay them and see how the structure has changed. Okay, if I can hand back to you, Wade, at this point to take us through the rest of the presentation.
Sure. Now actually handing off to Alice.
So as a relatively new starter to Schrodinger and from a predominantly ligand-based drug design background, there were a few challenges to adopting a predict-first mindset. I was first required to adapt to a culture change of a fast-paced collaborative effort. And this meant building comfort and confidence in the models being run, and also ensuring at the very least, I understand what the model is telling me in the most basic level, and that not to expect something that that model was never going to provide. I took time playing with live design to learn how to make full use of the tools at hand, and whether that's visualizing the data in the most effective way, or being able to run instantaneous docking and property analysis of my designs, as you've just seen. In doing so, it allowed me to appreciate the availability and the applicability of the models in each project. Ultimately, this led me to appreciate how differently we run drug design at Schrodinger. This cycle is very different from industry, where we don't just start at step 3 of designing and predicting, and we don't give up at step 5 if we don't yield expected results. Key to this is the medicinal chemists and the computational chemists working closely together, and not just underestimating steps 1 and 2. Ensuring that the time is taken to evaluate and define the domain of applicability of the models, and having open and honest discussions between the medicinal and computational chemists about what to expect from the model. These models shouldn't just be a magical black box. If data is not as expected, we use this to train and validate our models further and to improve and extend them. It might take one cycle, or it might take several cycles to get the working model. And it's the working model that is key to achieving a faster and more accurate design, make, test, analyze cycle. and ultimately, hopefully, leading to a safe, efficacious therapeutic.
And with that, I did want to end by mentioning that this is something that we've been thinking about at Schrodinger for a while, building a shared language between the medicinal chemists and the computational chemists, which is really important to allow for this cultural change and allow for this high level of collaboration between the two groups. And with that, we have invested in building online courses to really help medicinal chemists get more comfortable with fundamental concepts in computational chemistry. They can better understand what their computational colleagues are doing. And when working in a live design environment where some of these models run automatically for them, really understand how to best use them themselves and get the most out of the predictions, as Alice alluded to earlier. So our Introduction to Molecular Modeling and Drug Discovery course is a self-paced online course that really gives you hands-on introduction to industry standard computational tools, including live design, and allows you to really apply the principles that you're learning earlier in the course in a real hands-on case study. And over the last few years, we've had already 2,500 course alumni who have gone through this course, and really, and we're very proud of the ability to train, the next generation of medicinal chemists to become more comfortable with some computational topics. And with that,
thank you, everyone, for a great presentation. We do have a few minutes left for questions. Just a reminder to our audience, you can submit your questions in the Q&A box at the bottom of your screen, and any questions that we don't get to today will be answered by email after the event. So let's get to our first question. Here's one. How many people usually participate in the design club, and how many compounds get designed? We'd like to take that one.
Hi, I can take that one. So we normally have the whole department taking part in terms of medicinal chemists. So that's probably around 20 to 30 of us. But like we say, we open that out to all the computational chemists as well. And everyone can be designing in that session. And we can generate hundreds of compounds and design ideas. And then it's down to the team to then triage these design ideas and maybe separate them into the different motifs that have been designed. We can also leave the live reports open for anyone to access for another week or so, for them to go back in at their own time. So it's a really powerful tool for collaboration and for just generating more ideas within the groups.
Okay, thank you. Can you use live design to collaborate with external partners like CROs?
Yeah, I can answer that one. So we've got a couple of different workflows that allow you to use your own live design instance and separate live design instances to collaborate with CROs. It is dependent on exactly what information you would like to share with the CRO and how much input they need to have into the live report creation and the live report updating, but that is definitely something that we offer.
Okay, great. You mentioned FEP Plus as a powerful tool. Is this something you can run in live design as well?
I'll take this one. Yes, actually, and it's an awesome tool in live design. I was on a project where we had this installed, and we're running single edge, so we're comparing our new designs to basically one reference. But we get the results within hours once we start the job. So what I would do is I design about 100 compounds and let the jobs run overnight. And I knew coming in the morning that I would have some really nice, hopefully some really nice data for my next design cycle. So it really speeds up the design cycle to overnight from, waiting on a computational chemist to put everything together for you. It might be a couple days. So it's, really nice tool.
Great. Okay, next question. For the new hypothetical compounds, the properties which you are calculating, is it through thermodynamics principles or is it data-driven models?
Yeah, so we have both. We have the data-driven models, which could be QSR approaches or machine learning approaches. We've also got the physics-based tools. The models can be based on experimental data or based on predicted data. So yes, we can use all types of tools.
Okay. Great. Let's see here. After Design Club, who decides which compounds to actually make?
I suppose that would be down to the core team. So it's usually a team that approaches the Design Club for generation of these ideas. And as I mentioned earlier, that team will then look into triaging the sometimes hundreds of ideas that have been generated in this session. And then ultimately we wouldn't make all of those designs. We would then use all of our models to run docking, run FEP, analyze the ADME properties, and pick those that would hopefully give us the best balance of all of those to then go away and make.
Okay, great. Next question. How important is modeling in the initial validation of ligand interactions in the proposed binding pocket?
What we typically like to do is start with a crystal structure where we know the binding mode of the ligand. If we start with other structures, homology models or AlphaFold, for example, it's much higher risk. So we really like to look at cryo-EM or X-ray structures where we know how the ligand sits in the pocket. From there, we can use different modeling tools to understand better about the binding energies.
How do you compare the docking score and binding energy?
So we really use docking models as ways to generate high-quality poses. But really, when we're looking for a correlation with binding energy, we would look towards a tool such as FEP+. So we don't really look much at docking scores for rank ordering compounds, and really spend a lot more time using that to see will something reasonably fit in the pocket, will it form the interactions that we're hoping for, and in many ways using that as an input for the eventual FEP+ calculation.
Okay, thank you. You mentioned auto design module, but nothing was presented. Can you give some more details on what that is doing?
So Auto Designer is a tool for large-scale enumeration. So if we have an R group, let's say, on a particular compound that we want to generate millions or billions of compounds of analogs, we can do this using Auto Designer.
Okay, thank you. Let's see. What stage, if at all, the machine learning or other AI-related algorithms are coming up as relevant in your predictions?
So it can come up at various stages depending on how data rich or data poor what you're trying to predict is. So I think if you happen to have quite a bit of data on the project already, for some particular experimental endpoint, it's very common to try to build some type of model to see if I can help you with prioritizing compounds as the project continues. And then, as something that's actually used in the Auto Designer workflow is this active learning approach. After you have, so using machine learning models to help you to build off of physics predictions to allow you to then examine a much larger chemical space. So using both as at the level of individual property prediction and then also chemical space exploration.
Okay, thank you. Time for just a couple more questions. If one does have a protein crystal structure, have you used AlphaFold structures as starting points?
Like Jean mentioned, ideally we want crystal structure or cryo-EM. That would give us sort of our best indication of a true binding pocket. But there are some projects that have initially used AlphaFold whilst we're trying to get hold of crystal structures and cryo-EM. It's a very useful tool, but you have to be aware of its caveats.
Okay. I think that is about all the time we have today. Before we sign off, do any of our speakers want to share any final thoughts? Oh, we have one more question. Let's squeeze in one more. In the example you presented, you were really only altering one end of the ligand. In this type of case, could the whole process be automated? The docking seems so quick. Could you just scan millions of these compounds and pick out those that appear to have good properties?
So yes, I don't know that we want to do this within life design, millions of compounds at once. But yes, we can certainly import hundreds. I've done tens of thousands, let's say 10,000 compounds where I've imported to live design to run them in the docking. But as a visualizing tool, you don't want to bring in millions of compounds. It's really hard to digest that within live design. We usually, when we do look at millions of compounds, we do this all external to live design. We run the filters outside of live design, and then let's say we bring in the best 10,000, and then we can focus on those instead of doing everything from the outset with a million compounds in live design.
Okay, terrific. Again, do any of our speakers have any final thoughts you'd like to share? Nope. Okay.